Welcome to the last and final of the 4 sections of this Bio 201. We're going to head into the nervous system, and this is it. So your lecture test 4 is going to be over just the 4 lectures, 5 lectures associated with the nervous system. I did break this first intro to nervous system and nervous tissue into 2 parts, just getting some of the overview and general vocabulary out of the first one. And focusing on action potential and membrane potential and some of those things in the second one. So this first lecture is probably a little bit shorter, a little quicker. The second part of this the first chapter, chapter 11, the second part of chapter 11's lecture will be a little bit more intense. Our nervous system is the master controlling and communicating system of the body. So we're dealing with every thought, every action, every emotion, all of the cells, both electrical and chemical signals. And with the nervous system, your responses are fast and specific, and sometimes they're immediate. So you can get a quick kind of overview of what we're talking about and where it goes, which is pretty much everywhere. We have 3 major functions of the nervous system. Our sensory input, so that's the name for the gathered information. We get millions of sensory receptors to monitor both internal and external stimuli. We have integration, which is going to be our interpreting center. So it's going to interpret and process the information from the sensory receptors. And then it's going to decide what's going to be done from moment to moment. And then we have our motor output, which is the effects. Right? So the response to the stimuli. So we might activate effector organs like muscles and glands to cause a response. And that response is known as the motor output. Okay? So input, interpretation, output. Gather the information, decide what to do about it, and carry out that action. So the structural organization of the nervous system is 2 main areas, and then you've got some smaller divisions, and I'm going to show you a little bit more on the next slide. But generally speaking, we have our central nervous system, which is going to be the brain and the spinal cord. This is the integrating and control center. So this is going to be where we interpret sensory input and dictate motor output based on reflex, the current situation, and experience. The peripheral nervous system are your cranial and spinal nerves, essentially everything outside of your central nervous system. We're talking about nerves, so bundles of axons from the brain and spinal cord, and ganglia, which is a collection of neuron cell bodies. These are like your communication lines linking the body to the central nervous system. You have cranial and spinal nerves. We have our spinal nerves, which are impulses to and from the spinal cord. And you have cranial nerves, which are going to be information or impulses to and from the brain. And again, I'm going to tell you a little bit more about some of these divisions of the peripheral nervous system on the next slide. So I'm going to leave it at that here. We do have a few definitions of kind of what we're talking about when we talk about a nerve. So a nerve in general is a bundle of nerve fibers outside of the central nervous system. Those could be a mixed nerve, which is going to contain both sensory and motor fibers. It could be a sensory or an afferent, AFF, which is going to carry information from receptors to the central nervous system. And those cell bodies are outside the central nervous system. We could have a motor nerve, an efferent, EFF, right? So carrying out the effect, which is going to carry information from the central nervous system to the effectors. Those cell bodies are inside or within the central nervous system. And then we have our interneurons, which are just shuttles. So they're shuttling signals between those sensory and motor neurons. And they ultimately are the most abundant nerve in the central nervous system. They're kind of those relay signals. So they're pretty important. So this is the kind of the most simple way to look at the organization of your nervous system. So we obviously have the nervous system as a whole. And I defined for you on the previous slide your central nervous system, right? So your integrating and control center. and your peripheral nervous system, which is going to be everything outside of the central nervous system. The peripheral nervous system breaks down into smaller and smaller specific divisions with work going on. Okay, so we have two functional divisions of the peripheral nervous system. The first being the motor or the efferent, the effect system, and the other being the sensory or the afferent, the affect division. Your afferent division has to do with nerve fibers to the central nervous system from the body. So it's going to keep the central nervous system informed. You've got somatic or soma, meaning body. Those are impulses from the skin, the skeleton, the muscle, and the joints. And then you have visceral, which is dealing with impulses from the organs. So within your internal organs of the ventral body cavity. Okay, so somatic meaning kind of skin, skeleton, muscle, joints, and visceral, your internal organs. Your motor or your efferent, your effect system, right, is from your central nervous system to effector organs, so to muscles and glands to bring about the response. So your motor division is your response system. That division breaks down into two additional smaller sections, your somatic or your voluntary system, and your autonomic, your involuntary system. Okay? So everything here is from the central nervous system to effector organs. So somatic, being voluntary, is dealing with your central nervous system to your skeletal muscles. It's more that conscious control. And the autonomic or involuntary nervous system is dealing with the central nervous system to smooth, cardiac, muscle, and glands. And you're talking about getting that automatic response. We have two different parts to your autonomic nervous system, and they work kind of in opposition to one another. You have your sympathetic and your parasympathetic. And your sympathetic, fight or flight, parasympathetic, rest and digest. And we'll be talking about these in subsequent lectures as well. Our nervous tissue is made up of two types of cells. The first is our neuroglia or glial cells. There are 6 types of those, 4 in your central nervous system, 2 in peripheral. We're going to go over those next. Their job is to support the cells of the nervous system, right? So they're supporting cells. They aid in neuron function, and the neuron is the other type of cell. We have our nerve cells, which are the structural unit of the nervous system, and they respond to stimuli by changing membrane potential. We are going to talk in detail about the neuron and their role in a little bit later. But again, neurons or nerve cells are the structural unit of the nervous system, in which we respond to stimuli, and your glial or neuroglia are the supporting cells. So we have 4 types of neuroglia in the central nervous system. You can see them listed here. You can also see a nice picture of them. Your oligodendrocytes form the myelin sheath around axons in the central nervous system. So we're going to have one that does something similar in the peripheral nervous system. But this is your insulating and myelin sheath forming cell in the central nervous system. Your microglia are the defensive cells in the central nervous system. They monitor health. They phagocytize foreign material in the central nervous system, and they may move to injury sites. Astrocytes are your most abundant and versatile. They regulate passage of molecules between blood and brain, so they determine that capillary permeability. They support and brace neurons and synaptic endings. They're going to control that chemical environment around the neuron. They're the helper cells. And there's lots more, and it's listed in your book on page 393. They have very interesting ways of communicating between each other and all sorts of really neat things. And then your ependymal cells assist fluid movement in the central nervous system. So your cerebral spinal fluid, they're going to line central cavities of the brain and the spinal cord. Okay? So 4 neuroglia in the central nervous system. Only 2 neuroglia here in the peripheral nervous system. We have our satellite cells, which support cell bodies in the PNS ganglia. They're kind of similar to what the astrocytes do in the central nervous system. And then you have your Schwann cells, which are ones that we talk about more often as forming the myelin sheath around axons in the PNS. So they are similar to those oligodendrocytes, and they are vital to peripheral nerve fiber regeneration. So peripheral nerve fiber regeneration specifically. Here we are at the structure of a typical neuron. I've given you kind of that animated drawing and more of a micrograph picture. You can kind of see the difference there. So again, all of our neurons are going to respond to stimuli by changing membrane potential. And all of this talk about membrane potential is going to come in the next half of this lecture. So these are our nerve cells. Okay? They have 4 functions. They're going to conduct nerve impulses, that's excitability. Okay? They're going to live forever unless you damage them. So they have extreme longevity, lifetime, 100+ years. They are unfortunately functionally amitotic, so they do not have the ability to divide. So if you break them, destroy them, for the most part, you can't replace them. And then they're highly metabolic. So because of their high metabolic rate, they require constant oxygen and glucose, which is why it's so important to keep the nervous system healthy intact. So they're going to function in electrical signaling to generate action potential. We need that, right? Because if you think back to the muscular system, the action potential travels the motor neuron, arriving at that junction and sends neurotransmitters across so that the muscle even can begin the point of contraction. Okay? So we've got a number of structures here that we need to talk about a little bit. We have our cell body, which is known as the soma. Okay, so the cell body, or the soma, and you can see it on the drawing, contains a spherical nucleus and a nucleolus surrounded by cytoplasm, and you can see the nucleus labeled on the drawing. And it contains what are known as Nissl bodies. These are essentially the same thing as rough endoplasmic reticulum. Okay? If you have a cluster of cell bodies in the central nervous system, you're talking about nuclei. If you have a cluster of cell bodies and they're in the peripheral nervous system, they're known as ganglia. So make sure you understand the difference between nuclei and ganglia. Those are just clusters of cell bodies, and they're called something different whether you're depending on if you are in the central versus the peripheral nervous system. Okay, we have 2 types of processes that come off of that cell body. We have either a dendrite or an axon. Let's talk about dendrite first. Your dendrites are your receptive or your input area for stimuli. They're going to conduct that impulse toward the cell body. Graded potentials here, not an action potential. This is called a graded potential. We'll identify the difference later. And there could be hundreds of these branching per cell body. Okay? So again, input area. Okay? Then we have our axons, and our axons are going to send the impulse away from the cell body. They could be very, very short. They could be relatively long, and there's only about 1 per cell body. They are the conducting region of the neuron, and they're going to generate and transmit the action potential away from the cell body. They could have a lot of terminal branches, as you see in the picture, but again, only 1 axon. Okay? If you have bundles of processes in the central nervous system, those are tracts. If you have bundles of processes in the peripheral nervous system, those are nerves. So make sure, just like on the cell body, you know the difference between tracts and nerves for the axons, and again, based on location. A couple other things you see on this picture. You have an axon hillock. Well, it's not labeled, but it's essentially a cone-shaped area where the axon arises at the cell body. So where it says axon on the picture would specifically be the axon hillock. Then you have those axon terminals, which I already kind of mentioned. They're bulbous ends of an axon, and their job is to release neurotransmitters. Okay. We're going to talk about the node of Ranvier later. It's essentially a gap between areas of myelin sheath. It's going to create something we'll talk about in the next lecture, how the signal's essentially hopping across, skipping over those nodes, so we get an increased speed of conduction, and that's lovely. All right, so I've got some pictures here of the structural types of neurons for you. We have 3 different types. We're going to start with the picture on the very right, the multipolar neuron. The multipolar neuron has 3 or more processes, one of which is an axon from the cell body, and it includes motor neurons and interneurons. As a reminder, you're talking about shuttling signals and CNS to effector. So this is your major type in the central nervous system. You have bipolar neurons, which are very rare. They're really for special senses like the retina. You're going to have 2 processes, one axon, one dendrite, and it extends from the cell body. So these are kind of special, and it's truly just one of each, one axon, one dendrite. Then you have your unipolar neuron, and these are your fastest, but they're single short process that forms like the shape of like a T. They're primarily in the ganglia, in the peripheral nervous system, and you can see you get in this T formation, you have a distal sensory end, and at the other end is where it enters the central nervous system, and it includes things like dorsal root ganglion and all sorts of really interesting things. So these are your fastest, unipolar fastest. You might want to make note of that for quiz purposes. Hint, hint. And last but not least, for part 1, we're going to talk about the myelin sheath. So the purpose or the function of the myelin sheath is to surround axons in the peripheral nervous system by your Schwann cells and the central nervous system by those oligodendrocytes. We are insulating the axon in order to speed up impulse conduction. Okay? A little bit of protection, but really we're talking about speeding up impulse conduction. Myelinated fibers are going to be faster. Non-myelinated will be slower. Your dendrites are non-myelinated or unmyelinated. Okay? The node of Ranvier is that space that I showed you in that previous picture. And this is because your Schwann cells don't actually touch one another. So there's gaps of exposed axon between the myelin coverings. They're typically at pretty regular intervals. And this is a site for the very rare occasional axon collateral branching. This is a place where a single axon could branch. But the point here is to speed up this transmission where it's essentially hopping from one spot to the next. But the node of Ranvier is a place where there's a gap between those Schwann cells that wrap the axon. And you can see I've kind of started to hint at action potential and how that's going to work. And that is going to end part 1. That's just a lot of definitions. In part 2, we start focusing in on action potential and the membrane potential and how those phases work, conduction velocity, the difference between a graded and an action potential, or a graded potential and an action potential. And like the meat and potatoes of muscle tissue. So get ready for that. Hopefully this one was easy for a good start.